Rupa, do you have a background slide?
No.
Okay. Well, Rupa's talked before, but I'll just say that Rupa joined, she was in statistics working on her PhD, and she's joined our department about 2 years ago when she joined our program. She's working under the Maze Atlas project, which I think she'll talk about just for a second or two, which is a big multi-investigator collaboration funded by the USDA. And before that, she did a lot of work at ICRSAT. That's actually where she got her master's and was working in QTL mapping in that area a number of years ago. So,
thank you, Dr. Murray. So I'll be talking about what I've done so far as part of the Maze Atlas project, like Dr. Murray said. The project aims at understanding the genetics underlying adaptation of tropical maize in temperate environments, where the maize germplasm base is very low, narrow, and tropical maize has a lot of useful alleles and much diverse. But one of the major limitations of in adaptation of tropical maize in temperate environments is photoperiod sensitivity. So tropical maize is a short day plant, and when grown in temperate environments, it, among many other things, it flowers late, has excessive plant height, and is susceptible to common smut. So four major photoperiodic responsive genes have been identified and named Z-maze for photoperiodic response 1 through 4, and placed on the chromosomes 1, 8, 9, and 10. The positions of these loci have been redefined by analysis in nested association mapping population. So the genome architecture for my work is based on these two studies by Coles and nested association mapping analysis. So one of the major goals of QTL mapping is not just to place the loci on the genome, identify and place loci, trait-linked loci on the genome, but also to clone the causal genes or functional polymorphisms. And in general, the QTL are resolved to regions which are as large as 10 centimorgan. I'm saying as large as because this region, these regions usually contain many genes and require a substantial follow-up work. Nearly isogenic lines, or NILs, can be created across the targeted QTL regions to further narrow down the trait-linked region or fine map these QTLs. NILs are desired to understand the, to study the effects of these smaller donor fragments, smaller chromosomal segments within the targeted QTL, and they can thus be used to narrow down the QTL region. They're also desired to identify and separate the trait-linked, separate the loci which are in peak association of with the trait of interest, and that are linked to other traits, especially those traits that adversely affect the desirable phenotype. So to achieve this dissection, nearly isogenic lines can be developed in the context of near isogenic lines in allelic series. I can miss a pointer actually. In the context of near isogenic lines in allelic series, by making separate sets of these nils, or nearly isogenic lines, with diverse genetic donor parents. So in Nihilus, different donor alleles come from different genetically diverse donor parents at the same loci. So in the empirical study for this project, 7 tropical inbred lines from CIMMYT and IITA were used as donor parents, and 2 XPVP temperate inbred lines were used as the recurrent parents, and 4 nihilus for the 4 ZMPR QTLs were created, and each nihilus across the targeted region consisted of these 7 into 2, 14 genetic contrasts, and each of these set of nearly isogenic lines was created by crossing a donor parent with the recurrent parent, and the F1 was crossed to the recurrent parent, and using marker-assisted selection, introgressions were captured which were in the targeted region. And in further backcrossing generations, the donor fragments, the goal was to reduce the length of donor segments, donor introgressions, and also select for those individuals which had maximum recurrent parents genome in the background chromosomes. But in the process of creating this one set of nihilus, there are many questions that come up, and which are like, what should be the selection strategy? There could be many individuals with recombination in the targeted region. Which ones should we select to optimize the complete set of nearly isogenic lines? And what is the impact of the number of individuals in each generation? And how many backcrosses should be genotyped for each selected individual? And what's the optimum number of backcross generations? I'm trying to answer these questions using computer-based simulations, and that is what I'll be talking about today. So selection strategy is how to select and which individuals to select when you have, say, the backcross 1 individuals generated. So this has been done using marker-assisted selection and capturing recombinations that are near the targeted region, which is called foreground selection, or by maximizing the proportion of recurrent genome in the non-targeted areas, which is background selection, or a combination of both these strategies. And previous investigations have focused on introgressing 1 or more entire QTL regions. For our specific need, we wanted lines that had shorter donor fragment introgressions and not the entire regions. And also that these studies have worked on introgressing 1 or more QTLs simultaneously. We were not interested in simultaneous introgression of the donor fragments, but if nihilus is being constructed, or nearly isogenic lines are being constructed for a targeted QTL, then at the other 3 ZMPR QTL, we wanted to ensure that there was maximum recurrent parent alleles, so that the effect of these shorter regions on this chromosome can be studied. So we devised a selection strategy to suit our particular need. And for this, we first created an ideotype for this targeted region. Ideotype, it's not for this targeted region, but to capture recombinations. So this strategy was driven by the number of individuals that could be afforded to be genotyped. So say if we can afford to genotype 6 individuals, then the ideotype consisted of ideotypic nearly isogenic set, consisted of 6 lines, and we divided the targeted region into 6 equal length segments. It was the ideotype would be one where the donor alleles were here, here, here on these separate lines. But what might actually happen in say in the backcross 1 is an individual which has overlapping introversions. If you observe, this does not have overlapping introversion regions. So that is one of the criteria. So in backcross 1, there could be overlapping introversion regions, and for each of these ideotypic regions, base regions, independently and the best individual was selected with introversion as close as possible to one of the ideotypic boundaries, either on the left or the right, here on the left or the right. So using these as the first priority in selection for recombination breakpoints, and with the other criteria for selection, we had 3 levels. The first level was to select for those individuals that had maximum recurrent parent alleles on the targeted chromosome in the non-targeted areas. The second level, where we had recurrent parent alleles in these 3 ZMPR regions, and the third level where the rest of the genome was scanned for maximum recurrent parent genome. So we used this selection strategy to identify independently for each of these regions the best individual, to come up with a set of individuals that were optimum. So the next steps were to design the breeding strategies. So the investigators in this project, Dr. Randy and Dr. Nick, had a hypothesis that the number of individuals in the first backcross generation was probably very important and crucial in getting the desired recombinations. So we had differing sizes for the number of backcross individuals that were generated. And from each of these set of individuals, we either selected 10, 15, or 20 individuals, which is the number selected at each generation. And for each of these selected individuals, we created either 5, 10, 15, or 20 backcross individuals by backcrossing them to the recurrent parent. So we had 192 breeding strategies in total, using which we wanted to see the effect of the number of backcross progenies in the first backcross generation, and the impact that the number of individuals selected might have on the nearly isogenic set of nearly isogenic lines, and the impact of also the number of backcrosses per selected individual. So the validity, and I used simulations, as I already said, computer-based simulations to do all of this. And validity of any simulation is subject to how well the underlying biological process is modeled in the studies. So because the genetic variability in the lines was due to the recombination or the crossover process, the meiotic crossover process, we looked at how the crossover process could be modeled. So meiosis, as we all know, creates new combinations of chromosomes and chromosomal segments within a chromosome. And in prophase of the first meiotic cell division, crossing over occurs between the non-sister chromatids or the parental chromosomes, which creates a lot of variability, which is a source of variability. And when there are multiple chromosomes, multiple crossovers, any of the two sister chromatids participates in the crossover, irrespective of whether it was involved in other crossovers. So, which has been termed no chromatid interference. So we assume no chromatid interference throughout our modeling process. In the metaphase 1, the orientation of a homologous pair of chromosomes at the spindle equator is independent of how any other pair of homologous chromosomes is, which creates again a lot of variability in the final meiotic product. So in the process, independent assortment of these non-homologous chromosomes takes place, and chromosomes segregate in anaphase 1, which is followed by the meiosis 2. So there are a couple of stochastic aspects in meiosis, which are the number and positions of crossovers along the 4-chromatid bundle, the non-sister chromatids involved in crossovers, orientation of non-homologous chromosomes along the spindle in both meiosis 1 and 2, and finally the sampling of haploid gamete at the end of meiosis. And we simulated all of these aspects in our simulations, and one of the crucial aspects was the number and positions of crossovers. So to determine the number and position of crossovers, we looked at point processes, which are phenomena occurring at random points along in our case it's the chromosome, so I'm just calling it chromosome, but it's on the location or time scale. So point processes can be defined in terms of 3 equivalent terminologies. One is the crossover positions, which is in terms of the variable C, which says C1 is this position, C2 is this, C3, C4. And for modeling this, a continuous probability distribution is attached to C. The second definition is in terms of intercrossover distances, which is the distance between 2 crossovers. So these two are equivalent, which can be seen readily because C1, setting C1 equal to X1, we get C2 is X1 plus X2, C3 is X1 plus X2 plus X3, and so on. And these are equivalent. We can see that clearly. The third definition is in terms of crossover counts, where a discrete count distribution for n, the number of crossovers, is attached. And given n, the crossover positions are determined using a continuous location distribution. So this also is equivalent, or although they look like they're very different because C and X have discrete indices and they take continuous values, and N has a continuous index. Like n is defined as the number of crossovers until and including position s. So ns at this point will be 1 because that is the number of crossovers until that position here. So this has a continuous index, but it takes discrete values. So they seem like they are distinct random variables, but they can express the same events. Like for example, if you take the second crossover, the set of all these points, is the set of positions when the second crossover has not occurred yet. And this is expressed as set of all points such that C2, the value here, is greater than those values. And the same set of positions, which is this, can be expressed in terms of n as set of all these positions such that the crossover count is less than 2, where the second crossover occurred. So all these three definitions are equivalent. The one that we used for our simulation is based on intercrossover distances, and we followed a two-pathway model based on the distribution of intercrossover distances of late recombination nodules, which was done by them, Falk and group, on maze line. The first pathway was an interference pathway where the interference meant that if there is a crossover position that we determined at a point, then there couldn't be another crossover in the vicinity of it, which has a biological interpretation. Which is the biological interpretation of it. The distribution that was used for the intercrossover distances was gamma, and the shape parameter, gamma has two parameters. The shape parameter of the gamma represents the strength of the interference. The bigger the shape parameter, the stronger is the interference. So that was the first pathway with interference, and the second pathway was based on the second was a non-interference pathway, and the union of these two positions that were determined using both the pathways gave us the final positions of all the crossovers. So those are the genetic input parameters for my simulation, which came from many different studies. These are the shape and the shape parameters for the different chromosomes and the proportion of non-interference pathway 2 crossovers. That was important to create the non-interference crossovers. So all the simulations were performed using R programming language, and I used the package do parallel to implement parallel processing of simulation replications. So these are the number of crossovers on the selected meiotic product in using simulation methodology that I've described just now. So the likelihood of multiple crossovers being inherited was more for longer chromosomes, like red is the chromosome 1, black the chromosome 10, and the likelihood of multiple chromosomes being inherited was more for chromosome 10, and shorter chromosomes like the chromosome 10 were more likely to be non-recombinant and less likely to have multiple recombinations. So in my results, I've used chromosome 1 and 10 to compare how the genetic length might affect the results, which are also the largest and smallest chromosomes in maize. I'm still analyzing the data that I've generated using all these methods, but in general, the selection strategy was evaluated in terms of average donor introgression length of the selected nil set. So in the first backcross generation, like we expect the donor introgression length to be larger, and in subsequent generations, we expect that the selection strategy should select those individuals so that the average donor introgression length reduces. So that was one variable used to evaluate the selection strategy. And the sum of squared deviations in the introgressions from the idiotypic boundaries, because we were selecting based on the idiotypic boundaries, and so this gave us the amount of overlap, or whether the overlaps was reducing or not. And the depth of introgression, which we define as the number of times loci are covered by the individuals in the selected nearly isogenic line set. So the idea is that if a loci is covered too many times, we would ideally want that each loci in the targeted region is covered by 1, 2, or 3. But if it is covered by many individuals, then that is not what we desire. And these variables show four-way interactions, but I'm not happy with the graphical display of this. Like, for example, this is a graph for average donor introgression length, where red represents the values that I have from a population size of 50 in the first backcross generation, 150 in the first backcross generation, 250, 350. So this is like an overall look at how the mean introgression length is. It's obviously decreased a bit. And this is again a graph where this gray area is for 50, 150, 250, and 350, and the different colors here represent the generations. So with each generation, the mean introgression length is decreasing. But I would be happy to get suggestions from you to display for a graphical display of such data where you have all interactions, where you have the four-way interactions significant. Because there are 192 breeding strategies, and I feel that this looks really messy. I have also applied the selection strategy on the empirical data, and I still have to compare the selections made by using the selection strategy and the ones that Dr. Randy and Dr. Nick have already selected. And I also want to fit the two-pathway gamma model to the empirical data that we have. I want to thank you all for coming here and listening to me, and the Maize Atlas team, my graduate committee members, and others in my lab. Well, Bharat and John and Scott are not in my lab, but thank you. And this project, my work is supported by the Maize Atlas project. Thank you so much.
How small a crossover event can you detect?
You mean the length?
Yeah.
So in the first backcross generation, they were as big as 15 centimorgans. But I have to tell you that there are two ways I can compare. One is based on the markers that I have simulated, which are at discrete points, and the other is the simulated real situation, where we know the truth because we have simulated the genome. So I'm still not sure. I've not looked at the simulated real setup, but for the ones based on markers, it's in the first generation it's about and more than or around 15 centimorgans, and after that it kind of reduces to 10, 5, and so on. Basically it's 5, 2, 3, kind of something like that. And the targeted region that we have is 15 centimorgans.
I have to ask this question. So how many, and I don't know the answer, you obviously looked at a lot. How many per meiotic event, how many crossovers can you get in a chromosome arm like one?
So the gamma model had a shape parameter and a rate parameter. So the rate parameter is the one that kind of looks at the rate at which the number of crossovers happen.
And that's in a per meiotic event, not over generations. That's
No, it doesn't depend on the generations. It's more to do with the length of the chromosome. Like the way we have 1 Morgan, in a 1 Morgan region, theoretically we have on average 2 crossovers on the 4 chromatid bundle. That is how this Morgan is defined.
okay.
So on chromosome 1, which is about 2 Morgans, on average we would find
Find one chromosome, you want to find one crossover on each arm, right?
yes,
Okay.
yeah. So, I'm sorry.
But I mean, that's a really interesting question. So one of the things.
And what have they've looked at this? You can look at this with data.
Well, so that's the
Evidence of recombination on a meiotic basis. Do you get 2 crossovers in a single
So here's
chromosome arm?
here's the problem with this is a lot of people actually don't look at this. So there's been a few cytologists who have counted the number of recombination nodules, and the cytologists say recombination nodules are equal to the number of crossovers, although some of them are resolved as non-crossovers. Okay, so this one paper sort of tried to, it's been said it's the definitive model, and that's the Falk paper that she's using as a model. But I know that in my own analysis, when I was looking at the NAM population, my estimates are that there's probably about 60-some crossovers that are occurring per meiosis on chromosome 1. Okay? Remember, there's 4 chromatids, and only one of those is advanced. So you're only looking at one of those 4 chromatids.
I understand that, but
And we see 4 to 5 per generation on that chromosome. So most of those you're not seeing. The rest of them, they're occurring on those other chromatids. So that model fits the recombination nodules, but it doesn't necessarily fit the molecular data that we're seeing. And nobody's tried to really fit the molecular data that we're seeing. People think that recombination is well understood, and so they don't really, try and model it. Most people try and model it as, a recombination occurring between 2 loci and not occurring across the genome.
Yeah.
So this is really a whole in terms of.
But I'm just thinking practically. I mean, if you have a crossover on chromosome arm, do you get another one in that same chromosome arm in the same generation? That would have real implications for what you're doing because you're trying to break those.
Right.
Does that? I don't know. All right. I know in some crop species, for example, in alfalfa, you get one crossover event per chromosome. Okay, that's how it keeps it viable from an autotetraploid standpoint to keep viable seed set in normal meiosis. But I don't know in corn. I have no idea.
So if there is one crossover on one arm, then that is using the interference pathway. So the non-interference pathway is the one which takes care of another crossover. So in the empirical data, we have seen double crossovers in the targeted region. So the non-interference pathway ensures that there are recombination
So
counts.
people have physically found those types, double crossovers, in real.
Oh, yeah. Yeah.
In
But-
one
and-
generation.
And when she says, yeah, so when she says
Okay.
two pathway, so the non-interfer, so the cytologists, which don't interact with the modelers on this very well, shown that there's different genetic control over these two types of recombination pathways, the interference pathway and the obligate crossover. So maybe the alfalfa doesn't have an interference pathway. Maybe that's why you just see one. But, I'm sorry, I interrupted. You were going to say that. But that's the crossovers per meiosis, right?
Not per meiosis. This is crossovers in the selected meiotic product.
The selected.
That is the reason we have 0 to 7. If it were the actual number of crossovers, it would have been more than 7.
Right, but this is after one generation. This isn't the sum over 3 back crosses. This is, or is it?
Not sum of back crosses, no. This is one
One
generation.
generation. So the selected product, you'd see some chromosomes with 0, but some with as many as 7 crossovers on chromosome
Well,
1.
that actually happened only with chromosome 1.
Right.
None of the others had 7.
Because it does relate to size.
Yeah.
All right, let's thank our speakers again.
How you doing? Pretty good.